All right, welcome back, everybody. So we're going to finish up Chapter 2 that we weren't able to get through in class. So this is Chapter 2 is the Synapse. We had started talking about neurotransmitters, ion channels, started briefly talking about what happens when drugs interact with these receptors and neurotransmitters. So that's what we're going to talk about today, the latter half of Chapter 2. Talk a little bit more in depth about neurotransmitters and particular drugs that can affect these neurotransmitters at different stages. Yeah, I think that's going to be, let me see. Yeah, that's the majority of, very briefly, we'll talk about the endocrine system, just to set that up. So I think I had stopped talking on the, on dopamine. I am going to reiterate that just so that we have a complete in-depth conversation about the neurotransmitters. So let me go ahead and, where's a good spot? There you go. Okay, let's go ahead and switch over to PowerPoint. Okay, so I had mentioned this in class, the life cycle of a neurotransmitter, right? It first has to be made, first has to be synthesized, then it gets stored into vesicles, the little bubbles. I noticed on the homework for the brain coloring book, when I asked, color in the vesicle, you guys colored in the vesicle, but not at the very terminal button where the neurotransmitters are stored. So keep that in mind, even though vesicles are kind of traveling down the axon into the terminal button, the vesicle is basically being pumped up with neurotransmitters, right? All these little black dots here signifying the neurotransmitter. So usually that's done by a very particular neurotransmitter transporter, fills up these vesicles, and then we have that docking mechanism through calcium. Receptor binding, we'll talk a bit more about this. We had talked about two differences in postsynaptic receptors, meaning ionotropic and metabotropic. So ionotropic is going to be what you see here. Neurotransmitter binds, allows an ion to pass through. Metabotropic, the neurotransmitter that binds does not actually open that ion channel. It's coupled with a G protein that goes and connects to a different, basically a neighboring ion channel. And that G protein initiates a second messenger system. So indirectly, we also call it an indirect receptor, ion channel receptor, is the metabotropic version. But we're not going to revisit that today. Just wanted to reiterate that. But we will talk a bit more about different ways receptor binding can occur, and then of course reuptake, so recycling, by transporters that are found presynaptically, and destruction if that's going to be the case. And so this is important. This is what I was trying to get you guys to write about on the take-home quiz, is that drugs can interfere at every single stage here. They can either help or aid neurotransmitters and increase their activity at the synthesis stage, at the storage and release stage, receptor binding, or even at the reuptake. For example, when we talked about SSRIs, they were influencing serotonin here by blocking reuptake, which then allows the serotonin to continue hanging out in the synapse. Right, so keep this in mind, and we're going to go through some examples today, but the drugs can interfere at each single stage of this. Okay, I'm not going to go through each of the neurotransmitters. We're going to go through some in more detail. But we had our list here. Again, GABA glutamate, the main on-off, or rather off-on neurotransmitter. And this is how I prefer to show it. So glutamate and GABA, our main excitatory, our main inhibitory neurotransmitter, as well as glycine, which is a secondary inhibitory neurotransmitter, more important for peripheral nervous system, so spinal cord and lower brain stem. So these are all amino acids, and these are going to be your main activating or inhibiting neurotransmitters. Everybody else is considered modulatory, right? Acetylcholine, as well as our monoamines. So dopamine, norepinephrine, epinephrine, serotonin. The ones we're going to go in more detail are dopamine, which we did already talk about. We're going to talk about again. Serotonin. What were the other ones? I didn't think they were going to go that much into norepinephrine. Let's just walk through and see where we end up. Oh, acetylcholine. Acetylcholine is going to be one of the other ones. Okay, so dopamine. First of all, functional implications. What does dopamine do? Very important for, of course, reinforcement and reward. When you get a dopamine rush for something, means you're about to like get a reward. You're expecting a reward. That's when you actually get the biggest dopamine release. Mood, improved motivation, learning as well, attention and learning. So definitely some cognitive effects as well as movement, right? When we talked about Parkinson's disease, a lot of it was connected to an inability to initiate movement. So dysfunctions, Parkinson's disease, schizophrenia and bipolar disorder, as well as drug abuse. So drug addicts are going to have a dopamine dysregulation or chemical imbalance. And sorry if my voice is like weird and scratchy. I was delaying for a while of recording because of my cough in my throat, but hopefully this lecture won't be too bad. Okay, so we have not discussed neuroanatomy yet, but there are some regions that I want you to go ahead and start knowing because of their connection with creating neurotransmitters. So most of our neurotransmitters are going to be created in kind of this brainstem region, midbrain, or kind of here at the bottom of the frontal cortex. For dopamine, both are being made in midbrain, both pathways. One major area is the substantia nigra, so literally translates to black substance. And the other is VTA, ventral tegmental area. And as you can see here, you're not going to need to know right now where each of those pathways goes, but you can see that some will either go to kind of middle of the brain, center of the brain, kind of limbic system, basal ganglia system, whereas the other system actually kind of goes out into the cortex. So functionally, like I said, initiating movements. So that involves planning movements and controlling movements. Dopamine is essential for this. Learning memory and reward processing, right? When you're trying to figure out how something works, you get a big reward on like your trial and errors that work out well. You get that like, shucks feeling when like you miss it, right? So reward and motivation is closely connected to learning, of course. Planning, problem solving, lots of cognitive and attentional processes. Dopamine is pretty important for assisting that, or even kind of laying the foundation, functional foundation for that. Okay, synthesis pathway. So remember there are 4 stages. How is it first made? How is it stored and released? Receptor binding. We're not going to worry about receptor binding because there's usually a lot of nuances when it comes to receptors. We don't just have one dopamine receptor, we have multiple dopamine receptors, but for this class, I'm not going to ask you to remember those. So that's why 3 is grayed out. And then 4th, reuptake, either recycling or destruction, right? So for dopamine, we're going to start with tyrosine. Tyrosine is the precursor to dopamine. So precursor, think building block. Without this, you cannot make dopamine. Your brain cannot make dopamine. So we need tyrosine hydroxylase, which is an enzyme. Anytime you end on an ase, that's going to be an enzyme. Converts tyrosine to L-DOPA, and then L-DOPA is converted to dopamine by dopa decarboxylase, another enzyme. That's what you're seeing here. So tyrosine, L-DOPA, dopamine. And in fact, to get to norepinephrine, there's only one enzyme required that changes dopamine to norepinephrine. So these guys are biochemically related. Okay, when we're stored in vesicles, this is done by vesicle monoamine transporters. So if you remember back on our list, these are all monoamines: dopamine, norepinephrine, epinephrine, serotonin, and histamine. So for serotonin, we're going to see this again, that a monoamine transporter is responsible for packaging it into the vesicle. So vesicle monoamine transporter. If we're going to recycle it, it'll be by dopamine transporters. Different transporters, keep that in mind. This one, both are presynaptically, but one is intentionally trying to suck up all the dopamine from the synapse. This one is putting dopamine inside of vesicles. If we're going to break down dopamine and destroy it, that's going to be done by monoamine oxidase, MAO. Okay, and depending on where we are in the brain, dopamine can cause EPSPs or IPSPs, excitatory or inhibitory postsynaptic potentials. Please review these, EPSPs and IPSPs. I saw most of you in the quiz, in the take-home quiz, didn't actually talk about EPSPs and IPSPs. This is essential. When you have a neurotransmitter that binds on a postsynaptic receptor and opens an ion channel, allowing sodium, potassium chloride, or calcium. Did I say that right? Sodium, potassium chloride, or calcium, the 4 different ions, to enter or to exit the membrane. The consequence of those ions going in and out of the postsynaptic membrane, whether that's on the dendrites or the soma, are EPSPs and IPSPs, not an action potential. So what you get is neural integration. All those dendrites where you have synapses, and if they're synapsing on the soma, all of these guys are sending EPSPs or IPSPs. Neural integration sums all of that activity up. And if that activity is enough to reach the threshold of excitation, meaning -55, from -70 to -55, then you fire an action potential. Right, so that decision process is summing up all of the excitatory and inhibitory postsynaptic potentials together. If all of them are inhibitory, it's not going to fire an action potential, right? If there are enough strong excitatory ones, then you may reach the threshold of excitation. Okay. Oh, I feel like I'm easily out of breath. Okay, very briefly on Parkinson's. Like I said, we covered this in class, so I'm going to go through this pretty quickly. Neurological disorder, symptomatic of tremors. I'm sorry. Tremors, rigidity, poor balance, kind of shuffling, difficulty initiating movements. And this is because substantia nigra here, you can see, this is a healthy substantia nigra. Rather, I should say this is a healthy midbrain slice, slicing through the midbrain. This is obviously autopsy done, right? You can see this. You also find that with neurodegenerative diseases like Alzheimer's disease. You can see a lot of different things. Once the person has passed away and you look into their brain, you can see all these markers of it being Alzheimer's or Parkinson's in this case. But this right here would be the substantia nigra, where my red cursor is. So you can see that it actually appears black. In a Parkinson's patient, those dopaminergic neurons and cells are degraded, degenerated, and barely there anymore. So no longer that nice black dopamine-filled tissue. I might have to pause the recording in the middle. We'll see how this goes. So drugs to treat Parkinson's. This is the main one we talked about, L-DOPA. And L-DOPA, as you just saw, it's a precursor to dopamine. We talked about smell tests, so I'll skip over that for now. Okay, serotonin. Serotonin is implicated in a lot of things, not just your mood, right? When we talked about SSRIs, so antidepressants, the point is to increase your serotonin so that you're not depressed anymore. Improved mood, right? It's not just mood though, also sleep and dreaming, eating, arousal, and pain. So a lot of very basic biological functions that serotonin regulates. If we have a dysfunction, of course, in our serotonin, a serotonin imbalance, we're going to get things like mood disorders, eating disorders, sleep disorders, and anxiety disorders. So here, where is serotonin made? Primarily, it's going to be made in pretty much this entire brainstem region. So midbrain, pons, and medulla. Again, that's midbrain up here. Midbrain, pons. Pons is like the fat, like the belly, the fat belly part of the brainstem. And the medulla, the thing that starts down here merging with the spinal cord. So inside of here, you have what are called the raphe nuclei. And these are basically 9 major nuclei clusters of 5-HT. So 5-HT is a term that we use very frequently for serotonin, 5-hydroxytryptophan. 5-hydroxytryptophan. So anytime you see 5-HT, that just means serotonin. So these guys produce serotonin and then send it to wherever is necessary, whether it's down the spinal cord, the cerebellum, the cortex. But anatomical pathway, where does it start? The raphe nuclei. Okay, 5-HT. Serotonin synthesized from tryptophan. This is our precursor. Always keep that in mind. We have to start somewhere, right? So tryptophan is going to be what we start with. And in fact, you can actually get tryptophan from your diet, especially turkey. This is why Thanksgiving, when people eat all that turkey, and then afterwards you don't just have a carb crash, but you actually feel like sleepy and content. That's because you helped introduce some serotonin to your system. So tryptophan is your precursor. Tryptophan hydroxylase changes it to 5-HTP, 5-hydroxytryptophan, and then another enzyme, 5-HTP decarboxylase, changes it into serotonin, 5-HT. So similar to dopamine, we're going to have a precursor, a midway transformation point, and our end result, serotonin. Stored, same as dopamine, by vesicle monoamine transporters. And then reuptake, same as dopamine. We're not going to have dopamine, but serotonin transporters broken down by MAOs, which are monoamine oxidase. Monoamine, let me look real quick. You have to cheat. Oxidase, monoamine oxidase, MAO. Okay. All right, so that was serotonin. And we'll talk about drugs that affect serotonin in a second, but let's get through the rest of the neurotransmitters that I want you to know in more depth. So acetylcholine, functional implications, motor function. So this is going to be motor function primarily for just really most of your movement of your muscles, especially peripherally. Right, dopamine was for initiating motor movement. This is for pretty much the rest of your motor movement. Learning and memory, if we're looking CNS cognitively, going to be very important for learning and memory, as well as motivation, similar to dopamine. And when it comes to sleep, very particularly REM sleep. So dysfunction in acetylcholine, some of it, of course, will be some sleep disorders, as it does play a role here in REM sleep. Epilepsy, if we have too much acetylcholine spouting out in your system, epilepsy, of course. Alzheimer's disease, some some drugs to help treat Alzheimer's disease are acetylcholine, basically help increase the activity of acetylcholine. And another dysfunction that is also motor related is myasthenia gravis. Here, where acetylcholine is made, primarily produced, is going to be in the basal forebrain. Basal forebrain, also called the nucleus basalis. It's basically going to be, if you bend your brain, where your frontal cortex kind of bends over, there's going to be a little part here where it's like packed away in a little niche. This is basically like almost right above your eyeball. This is going to be the nucleus basalis or the basal forebrain. This is where most of your acetylcholine is being made. So as I mentioned, REM sleep, activation of the cerebral cortex, facilitating learning, right? It's a modulatory neurotransmitter, just like the other ones we've been talking about, but this one is a more activating neurotransmitter. Typically, it's going to be more on the side of excitatory rather than inhibitory. Also helps controls rhythms of the hippocampus. Like I said, I know we haven't done neuroanatomy yet, but hippocampus we'll find is essential for being able to form memories. Hence, Alzheimer's disease, we're having a dysfunction with our memories. Okay. Synthesized from two different precursors, from choline and acetyl coenzyme A. So here, choline as well as acetyl coenzyme A. Choline acetyl transferase basically helps put these two together so that we get acetylcholine with the side product of coenzyme A. So it's a little bit different than our other neurotransmitters. Also, the destruction is going to be a little bit different. So it's broken down by acetylcholine esterase. We briefly talked about that. One single acetylcholine esterase can destroy 5,000 acetylcholine. And what's interesting when it comes to recycling is that actually only the choline molecule is recycled into cholinergic cells. So not the coenzyme A part or the acetylcholine enzyme A part. As far as storage goes, vesicle acetylcholine transporters. These are usually pretty straightforward. It's going to be transporters that are specific to that neurotransmitter or that type of class of chemicals, so monoamine transporters. Okay. All right, let's move on to drugs. So when we talk about a drug having a psychoactive effect, it's because it's affecting the neurotransmitters that are responsible for making sure everything is communicating properly. And most of the time, drugs are going to be doing this by interfering with the synapse communication, right? I told you those 4 different locations, the synthesis, the storage and release, the receptor binding, and then the reuptake and destruction part. When we talk about a drug, it can either be classified as an agonist or an antagonist. So an agonist is if a drug helps facilitate the effect of that neurotransmitter. So for example, if you're depressed and you take an antidepressant such as an SSRI is there to help facilitate serotonin, right? Your brain doesn't make enough serotonin, so it does this fancy thing where it like blocks the reuptake so that it hangs out in the synapse longer. That SSRI is going to be a serotonin agonist because it's going to help facilitate the effect of that neurotransmitter. Antagonist is the opposite. It either blocks or inhibits the effects of that neurotransmitter. So I mentioned SSRI here because it's easy to think, oh, it's called selective serotonin reuptake inhibitor. So it inhibits, right? It means it's an antagonist. No, you have to consider what is it doing overall to the neurotransmitter? And SSRI overall is going to help convince the synapse that there is more serotonin than there is, right? Because serotonin continues to hang out, it continues to bind, new receptors are written, and then you have improved mood because there are more spots for what little serotonin you have to bind and have that mood enhancing effect. So an SSRI is an agonist, serotonin agonist. And we'll show a few examples of each of these for the different neurotransmitters, the main ones we talked about. Also, one thing to keep in mind, most drugs actually have more than one effect. So this one here is a pretty stark illustration of that for morphine. So we'll use morphine to not feel pain, like after surgery, for example. So a very low dose of morphine is already going to have analgesic effects so that you don't feel pain anymore. However, if we move the dose of that drug to a higher degree, it's not just affecting your pain perception, it's also going to end up affecting your respiration. So too high of a dose is actually going to have a depressive effect on respiration. So if you want to be careful, for example, with morphine, that you don't do too much or you stop breathing, right? Just enough so that you don't feel pain, not too much to where you kick the bucket. And generally, that difference between the effectiveness of that drug, the reason you're using it, and it being lethal or toxic is called the margin of safety. Ideally, you want a very wide margin of safety, right? A little bit of that drug's going to do its thing. If you accidentally overdose, it's not going to be at a lethal or toxic level. You have a wide margin of safety before it affects you ill, so to speak. Okay, just to reiterate the different locations that drugs can have their effects. So we're going to go through these. So starting with neurotransmitter synthesis, we talked about one example of this, L-DOPA, right? So you can give a drug, which is L-DOPA, that's given to Parkinson's patients. Their substantia nigra is degenerated. They cannot make new dopamine themselves, but what you can do is you give them a precursor. L-DOPA is a precursor. Once that precursor actually gets into the terminal button, this L-DOPA passes the blood-brain barrier, goes into the terminal button, and then enzymes are hard at work and making a new neurotransmitter. So this is how you can give Parkinson's patients the ability to make their own dopamine. So because you're adding a precursor to it, this is going to be an agonist. You're help facilitating dopamine production, right? Dopamine action. So most neurotransmitters are produced from precursors. Some drugs can act as precursors. And if they do, and if you give that drug, then this increases the amount of neurotransmitter by that cell, right? Like L-DOPA. So in blue are going to be agonists, and red are going to be antagonists. All right. When it comes to reuptake or destruction, I guess I'm jumping to the fourth area. One we talked about was SSRIs. This is another one related to dopamine. So cocaine, right? Let's say dopamine is in your vesicles here. It goes and binds and then dumps out into the synapse, and then dopamine goes and binds to a receptor. And then once it's done, it's like, all right, I'm done. What do I do now? This little transporter will basically suck it up, and recycle it again. Cocaine, however, is going to block that reuptake. And because it blocks the reuptake, dopamine continues to hang out in the synapse. It can go bind to a receptor again. So just like with an SSRI and serotonin, this is going to end up artificially enhancing how much dopamine is in the synapse, right? So it's going to be an agonist because it enhances the effects of dopamine at this synapse. So terminal membrane transporters, so terminal, axon terminal, terminal button, terminal membrane transporters, they're responsible for recycling or reuptaking neurotransmitters. Drugs can inactivate these transporters. If a drug blocks reuptake of that neurotransmitter, it ends up enhancing the presence of neurotransmitter in the synaptic cleft. So those are two dopamine ones, L-dopa and cocaine. Both are agonists to dopamine. Work in two different ways at two different parts of the synapse. Okay, so these are three different, classes of drugs that I want you to know for dopamine. So L-dopa, we talked about this. Amphetamines, methamphetamines, cocaine, and methylphenidate, which is Ritalin, they all do the same thing. They all block reuptake of dopamine, as well as norepinephrine, actually. So if you block reuptake, it enhances the effect, hence agonist. I want to introduce one antagonist here. And later in the semester, we're going to talk about psychiatric disorders more. But chlorpromazine, this is a dopamine antagonist. So it actually blocks the ability of dopamine to properly bind to postsynaptic receptor. So don't worry about D2. Like I said, there's different types of dopamine receptors, but this is a dopamine receptor antagonist. So people who have schizophrenia or bipolar disorder with psychosis have too much dopamine in their system. So hallucinations, delusions, right? So what you want to do is you give them, for example, chlorpromazine, and this will reduce the amount of dopamine in their brain because it will not allow dopamine to bind as frequently to the receptor. All right, and we already went through this, so I'm not going to do this one again, but this is walking through the SSRI. Sorry, my nose is like... Okay, serotonin. We have a few serotonin agonists, so something that enhances serotonin. These are the ones that I want you to know. LSD. LSD is a direct agonist of postsynaptic 5-HT serotonin receptors, producing hallucinogenic effects. Antidepressants, SSRIs, we talked about this, how they work, so definitely know that, as well as ecstasy, MDMA. It blocks and reverses serotonin and norepinephrine transporters, right? The thing that would help you recycle it, they block it, enable norepinephrine and serotonin to continue hanging out in the synapse. Too much of this, though, can end up result in neuronal damage and cognitive deficits. So no antagonists to know for serotonin, only these three different agonists. Okay. Neurotransmitter storage and release, right? So back here, we have a precursor. The precursor is the building block, builds it into a neurotransmitter, and then the neurotransmitter has to get packaged into a vesicle. And at some point, when an action potential comes down, that vesicle comes down, binds to the membrane, and dumps the neurotransmitter. So there are two drugs that I want you to know for acetylcholine that will affect this system right here. The first one is black widow spider venom. Black widow spider venom affects acetylcholine, and it does this by stimulating the release of this neurotransmitter. So any vesicle that is stored here inside of the terminal button, remember I told you this can be sometimes hundreds to thousands of vesicles. If you get bit by a black widow and that venom seeps into your system, it will trigger all vesicles stored with acetylcholine to immediately go and dump their contents into the synapse. So synaptic vesicles, they dock with proteins on presynaptic membranes for release, right? We talked over that with calcium. When calcium enters the cell, it enables that docking mechanism, and then it tears it open so that this neurotransmitters can come out. Drugs can activate these proteins, just like the spider venom here. If that happens, then it increases the amount of neurotransmitter released by the cell. It's because it's dumping neurotransmitters into the synapse. So black widow spider venom causes flooding of acetylcholine into the synapse. This results in severe muscle spasms, contractions, right? Once all the acetylcholine is used up, then it's followed by paralysis because all the acetylcholine was used up. Now there's nothing left. So basically after seizing, and then you're going to have paralysis, including your heart, which is a muscle. Your heart's going to seize up too. Okay. So because it's enhancing the effects of acetylcholine, black widow spider venom is a agonist. Now we also for acetylcholine have an antagonist, which is botulinum toxin. So here, botulinum toxin actually inhibits the release of the neurotransmitter. So even though these are filled with neurotransmitters, this binding mechanism isn't going to work. So drugs can deactivate those proteins that allow that docking to happen. This decreases the amount of that neurotransmitter because no fusion happens. Even if calcium comes into the cell and says, docking is ready, that vesicle, wants to go dock with that other protein. If you have this drug present, for example, botulinum toxin, it's not going to allow that docking to happen, so no release of neurotransmitter. So it's impairing or blocking or inhibiting this particular neurotransmitter, acetylcholine, to properly work. And botulinum toxin is what's in Botox, right? So Botox prevents the release of acetylcholine, and this is how it works, by relaxing your muscles. Basically, mild muscle paralysis, right? So smoother skin, less wrinkly because no contractions are happening, because no acetylcholine is coming out. So by blocking acetylcholine, it relaxes the muscles, right? Okay. Now, what I said is that most drugs, and that's still the case, are actually going to be affecting these receptors, the postsynaptic receptors. I'm going to walk you through, I think, just one example, though, which is nicotine. So when you smoke cigarettes, nicotine will help stimulate acetylcholine. Where normally acetylcholine would bind to, nicotine can take that place and stimulate those ion receptors and open up the ion channels. So neurotransmitters typically bind to postsynaptic receptors, right? Drugs can mimic these neurotransmitters. If they are mimicking the neurotransmitter and binding on the same spot that neurotransmitters would, we call these a direct agonist. It activates the receptor, opens the ion channel, and then EPSPs or IPSPs happen. So nicotine binding to nicotinic acetylcholine receptors, this is where the stimulant effects of that drug come from. Okay, so for acetylcholine, we talked about a few drugs. These are the drugs I want you to know for acetylcholine. Black widow spider venom stimulates the release of acetylcholine, just basically dumping all of acetylcholine in. So it's an agonist, as well as nicotine, a direct, and I'll talk about this in a second, this word here, direct agonist. But it's a direct agonist for ionotropic acetylcholine receptors. An example of an antagonist is Botox. It prevents the release of acetylcholine. Okay, so this is when a drug binds to a postsynaptic receptor, it can do that in two different ways. Either a drug is going to bind at the same location that a neurotransmitter normally would, like nicotine, right? Normally acetylcholine would go bind, open up the channel, but if you have nicotine in your system, it's going to go bind on that spot instead, of acetylcholine, and then nicotine can open up that channel. This would be referred to as a direct agonist or a competitive receptor binding. Competitive because it's competing with the neurotransmitter. We have another way that drugs can do that too. So this is one way. Either it directly competes in a direct way with a neurotransmitter, or there's an alternate binding site. So this was what I was showing with my bell peppers. All of these bumps here, these are different binding sites for ligands. Neurotransmitters are ligands, and drugs are ligands as well. So here, for example, a neurotransmitter might bind on this site, and a drug can bind on another site. These are not going to compete with each other, so non-competitive, and this is also indirect, right? If that drug comes and binds to the alternate site and it opens up the channel, then it is an indirect agonist. We can also have a direct antagonist. The drug comes on the competitive site for the neurotransmitter and doesn't allow this channel to open. It'll be an antagonist. Or on the alternate binding site, a drug comes, binds, and despite the neurotransmitter binding, it will not open it. It'll be an antagonist. So basically, you can be a direct or indirect agonist or antagonist. Okay. So here I wrote it out for the indirect portion. Indirect antagonist, a drug attaches itself to an alternative site and prevents the ion channel from opening. Or you can be an indirect agonist. A drug attaches itself to an alternative site and it facilitates the ion channel opening. This is actually how Valium works. It's an indirect agonist that requires concurrent. So just like here, you see a neurotransmitter and the drug. This would be Valium. Sorry, it's an agonist, so let me put it over here. This is your drug, this is your neurotransmitter. This would be Valium, and this would be GABA. So GABA is going to be your main inhibitory neurotransmitter, right? So if you have GABA binding and you have Valium binding, it's going to enhance this effect, keep the channel open longer so that more chloride ions can come in, hyperpolarize the membrane to where the next neuron is less likely that the next neuron is going to fire an action potential. So IPSP, inhibitory postsynaptic potential. If it's going to help not fire another neuron, this is how you get the anti-anxiety or anxiolytic effects of Valium. It reduces the brain talking to itself so much. In fact, for GABA, normally I put a slide up on here that talks a little bit more about it. But GABA has multiple binding sites. So you might have a GABA binding site. You might have a benzodiazepine, which is what Valium is, benzodiazepine. You might have another one for a steroid, and another one for barbiturates, which are old-fashioned anti-anxiety drugs. So this is why if you were to ever take an anti-anxiety drug like Valium, Klonopin, Xanax, all of the benzos, it has a warning on there that says do not drink alcohol excessively when you're taking this, because guess what? There's a binding site for alcohol on the same receptor. So if you have GABA and Xanax and alcohol all binding together, that's massively going to depress your brain, right? And just like I talked about with morphine, it could massively depress your brain where it also starts affecting other things like your breathing, like properly being able to regulate your blood pressure, stuff like that, right? So keep that in mind. When we have these alternative sites, it can also, if you're not careful with that drug, cause problems. So that's often where those warning labels come from because of the side effects that it can cause. Okay. I highly recommend, by the way, for the exam that you study or just re-listen to what I was just saying about GABA receptors, GABA and its alternative binding sites, because that's probably something I will definitely be asking about. on the exam. So make sure that you revisit what is GABA and what are these indirect agonists. Okay. All right, one last kind of odd mechanism there is. So we talked about this here is a presynaptic terminal button. This is going to be your postsynaptic cell, right? Postsynaptic membrane with a whole bunch of receptors. Once the serotonin, in this example, serotonin comes out, it'll get recycled by transporters, right? This is the mechanism that we've been talking about so far. Now, we also have what are called autoreceptors, which are basically receptors that are on the presynaptic terminal, the terminal button. And autoreceptors actually help regulate their own neurotransmitter release. So this guy is having an influence on how much serotonin is being packaged and released. It's kind of like a brake system for yourself. Generally, if an autoreceptor is stimulated, so something binds to it, it's stimulated, it's typically a signal of, hey, slow it down, right? So if you stimulate an autoreceptor, it's going to impair serotonin release. So you can think of the stimulation of autoreceptors being antagonists to the neurotransmitter that it's regulating. It's just a more nuanced way of regulating your neurotransmitter. Autoreceptors are generally metabotropic, and like I said, in most cases, they're going to be inhibitory in nature. Okay, I think we're nearing the end here. Yeah. So other chemical communication things that we have other than neurotransmitters, we can have neuromodulators, so peptides. These work a little bit different. They can travel further, are more dispersed. They kind of have an area of effect that they influence, a more widespread activity. So for example, endogenous opioids, something our body produces to help us deal with pain. This is going to be more widespread activity, not highly regulated by a synapse. We can also have hormones. These are secreted by cells of the endocrine glands. I'll show that on the next slide. A lot of different chemicals can function as hormones and as neurotransmitter. As an example, norepinephrine and epinephrine is the hormone version is adrenaline and noradrenaline, right? So depending on where you are in the body, they can function as hormones or as neurotransmitters. Many neurons actually contain hormone receptors. So proper hormones can actually go and influence brain activity by binding to those neurons. Sex hormones can change behavioral states. Yep. Peptides, second messengers, yeah. So basically, neurotransmitters are not the only ones. Hormones can have a pretty strong effect as well on brain communication. So endocrine system, and like I said, again, I sometimes will do neuroanatomy first before we get into the nitty-gritty stuff. But your endocrine system are basically your hormone system, right? So you have your thyroid glands, adrenal glands, and liver, kidney, all of your organs are making different hormones to help regulate your sex hormones, whether it's in the ovaries, placenta, or testes. And then we also have up in the brain, we have 3 main locations for hormone production. The hypothalamus, which is this guy here, sorry, not the red one, this right here, where my red cursor is. This is the hypothalamus. It is the main, master regulator of all of your hormones. Then we have 2 additional glands. We have the pituitary gland and the pineal gland, which is going to be back here. Yeah, it's hard to see, but it's going to be kind of like somewhere in the back of the brain here. Pineal gland is for sleep hormones, so melatonin. Pituitary, which is here, this is referred to as the master gland, which really, and I'll show you that on the next slide, produces a whole ton of different hormones. Some of these hormones will travel to different areas of your body to communicate, for example, the HPA axis, and we'll study at a later date the HPA axis, but that's the hypothalamic pituitary adrenal axis, right? So the hypothalamus via the pituitary sends a signal to the adrenal glands to make cortisol, right? So you can see here, anterior and posterior pituitary make all of these different hormones. Some of these are going to be sex hormones, thyroid hormones, growth hormones, oxytocin, all that kind of stuff. And hypothalamus is basically regulating everybody, has a variety of its own hormones that it releases and controls. And then the other central nervous system one is the pineal gland. This releases melatonin, right? So it helps you with sleepiness. So don't worry right now about learning all of these guys. Obviously, some are going to be familiar to you, like insulin, cortisol, testosterone, estrogen, leptin. But I'm not expecting you to know this entire list right now. What I want you to know is the three central nervous system ones: hypothalamus, pineal gland, and pituitary gland, but we will go into more detail at a later date. And then just generally what the endocrine system is. It's your hormone system, right? Many different organs need to be regulated, and the way that communication happens is through hormones, not neurotransmitters. Okay. So that wraps up the end of chapter 2. So a few thought questions, I'll leave you with these. Describe the structure of the synapse. Really, I highly recommend that you honestly draw this out with all the changes that have happened in this class because of not being able to meet in person all the time, and now potentially a hurricane coming through. I normally at this point would have given you a homework to kind of sketch out yourself the synapse, but I think I'll wait maybe for a later date for that. But think of, all the different elements that you learned presynaptically, postsynaptically, autoreceptors, vesicle docking, precursors, transporters, 2 types of transporters that we learned. What terminates a postsynaptic potential? See if you can answer this one. And please, please, please make sure that you review this. What are EPSPs and IPSPs? Make sure that you know the difference between an action potential and an EPSP and an IPSP. Okay, so I think that was chapter 2. So I will see you guys. I'm uploading this tonight, which is Sunday. So I will see you guys tomorrow then, I believe. And we're going to do review for the exam. That was the last slide, right? Yeah. Okay. So yeah, we're going to do a review for the exam. And then depending on whether or not we have class on Wednesday, if we do have class, we'll just continue with the material. So chapter 3, that is not going to be on the exam, but just so that we can move forward with our material. Obviously, if we don't have class, then we don't have class. And I will record that lecture for chapter 3. But nevertheless, I moved exam 1 to next week. So not, what is the exact date? I think it's October 3rd. Double check the announcements. But yeah, that was the wrap up. This lecture was the wrap up on the last of the material that you need to know for the exam. Okay, all right. As always, if you need to email me, if you have questions, office hours, I'll be back in full swing starting tomorrow. So if you need to come by office hours, please do. Please talk to me. I know that already things are getting complicated. Please take advantage of office hours. I'll be wearing a mask. Don't worry. But it can help if you have someone walk you through it, right? So please take advantage of that. Okay, I will see you guys tomorrow. Bye.